Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Today on the Microscopist, I'm joined by molecular biologist, a biophysicist and Nobel Laureate Richard Henderson of the MRC LMB down in Cambridge. And we'll discuss some of his pioneering work in the field of electron microscopy and ask him why understanding the structure of a protein is so fundamentally important.
The proteins do everything in biology, and we need to know their structures.
And as a physicist, what attracted him to biology and biophysics?
Biophysics needs physicists to do physics in biology.
We'll talk about his motorbike adventures in Scotland.
It turns out riding a motorbike or a scooter in Scotland in the winter,
It's not very clever,
it's a dangerous thing, actually.
Watching TV and his newfound passion for Netflix.
The one I can strongly recommend and I thought was wonderful was the series called Breaking Bad.
All in this episode of The Microscopists. Hello and welcome to this episode of The Microscopists. I'm really excited today because I'm joined by Richard Henderson from the MRC Lab of Molecular Biology. And actually, I had a very quick look, Richard, at your CV, but not too much detail, so I'm looking forward to learning more. But what was amazing is you are at the MRC of Molecular Biology. You got your undergraduate in physics at the University of Edinburgh, and you got the Nobel Prize for chemistry. Is there anything you don't do?
Well, most things I don't do, actually, but when Joachim Frank and Jacques Dubochet and I went to Sweden to get this Nobel Prize, there's two funny stories. One is we were all told somebody has to give a 2 to 3 minute speech thanking the king for giving us the Nobel Prize. And so I decided the best thing was to remain silent. But after about a couple of months, an email came from Joachim Frank saying, "Dear Richard, I notice one of us has to give a speech. I think it should be Jacques Dubochet." So then I sprung into action. I said, "Excellent idea. Why don't you ask him?" So Joachim then asked Jacques whether he'd be willing, because he was the most charismatic Frenchman, to give the speech. And Jacques then replied, he said, "In these Nobel Prizes where there are 3 people sharing," he said, "normally it's the oldest person who gives the speech, which would be Joachim Frank. Or failing that, it should be the youngest person, which is me." He said, "Never is it the middle person." So at this point, I could see these dangerous waves coming in from the side. So I sprung into action and wrote a very long, persuasive email to Jacques about his character, his charming nature, his charismatic speech, how it, he's the only one who could do it. And he replied with counter arguments to all of my arguments. But then in the end, and this was his mistake, he said, "Of course, but I am a Democrat." And so, since we'd already arranged it, Jacques had to give the speech. And he said exactly what you said. He said, "How is it that 3 people from physics working in biology get a Nobel Prize in chemistry?" And so when Jacques told us this is what he was going to say, I said to Jacques, "You've heard of the Peter principle, which is that in any organization, you get promoted every time you can do the job well until you get your final promotion into a job that you're unqualified and incompetent at. And then you never get promoted again. So you remain in this job. So the equilibrium situation in the world is everybody's promoted until they're incompetent. So I suggested to Jacques, this is what he should put this in his, and he did actually, he put it in, but then he said, no, he didn't really agree with this. He thought that it showed that science nowadays was completely unified, which was a kind of nice way to end, actually.
Yep.
Anyway, so that's the reason, is that it's cross-fertilization, biology needs physics, and the chemists are the most gregarious, outgoing, and welcoming, because they've realized, the periodic table's finished, chemistry isn't finished, there's still lots of things to do. But if you look at the chemistry department, probably in York or in Cambridge, it's full of people doing biology or protein or nucleic acids and things like that. So the chemists are very, very good at being like the Catholic Church. They welcome everyone.
Yeah. And as biologists, we need chemists.
Indeed, biologists need everybody.
Yeah, it is. We kind of the end point, the physics, the development of all the physics, the engineering, the chemists are all there to really help us to answer a question.
That's right. Yeah. Well, in the end, of course, in biology, 50 years ago, there were no genome sequences and possibly one protein structure. Now we have millions of genome sequences, and apparently in the protein data bank there's 170,000 structures now. So pretty soon you'll have the genomes of all the animals that people know the name of, like giraffes, elephants, lions, tigers. All the genomes are done, and you'll have protein structures known, maybe not for every single protein in every single species, but at least a representative example of them. So really, biology is reaching the stage where a hell of a lot is known, but there are millions of untapped areas. How the brain works, how how developmental biology of all the different tissues and organs. It's really a big, big open book for quite a few years to come.
So I was going to ask you one of the. I've just got a few questions I was going to ask, and so for the non-specialist that's actually watching or listening, why is the structure of a protein so important to actually understand?
Well, it's difficult now in the modern world to put yourself back to the time when you didn't know protein structures. But, back in the 30s, they were thought of as unstructured colloids. And actually, until it was shown that each protein was unique and had a unique sequence, because, in 1950, when Fred Sanger was doing his insulin sequence, it's not many amino acids. Many people didn't believe it until that was done. But now, of course, we know a lot more than we did in 1950. And you know that almost every reaction in biology is catalyzed by an enzyme, and all enzymes are proteins apart from a few that are RNA-based catalysts, and they're not very good catalysts. Proteins are much better. So basically, any function in biology, chemical catalysis, mechanical work, motors, recognition, signaling across membranes, signaling, by diffusion, it's all based on the ability of individual proteins to do particular reactions. And you need to know the structure and how they work for all of these proteins. And now, of course, we know a lot about a lot of things. We know about muscle contraction, oxygen in the blood, about all the receptors, about adrenaline and your heartbeat, and, how nerves work and signal, and they're all based on proteins. So each protein has its own interest, actually, and lots of people used to spend their whole life working on one. See, Max Perutz worked on hemoglobin for his whole life, and some of us tried to persuade him to be interested in other things. But in the end, he developed a slight interest in Huntington's disease from these polyglutamine tracts when he was older, but he was still mainly focused. But now, students can determine many protein structures in their PhD thesis because of the power of the methods. But anyway, the proteins do everything in biology, and we need to know their structures. But once they're known, not everybody needs to know everything about proteins. You probably need an undergraduate degree. You need to know a few examples, and then the general principle of it just feeds through into subconscious minds of the scientific community and the lay community. I've not worked on all that many proteins, but even now there are new ones being discovered, or there are proteins that are in certain peculiar, let's say amoeba or bacteria that haven't been found by homology in other species. So there's still an untapped area with proteins that are popping up out of the undergrowth. But generally, we know now a lot about proteins. So it's actually not quite a closed book, and it's not like the periodic table, but it's getting close.
And of course, so once you know the structure, the importance of knowing it means you can then direct drugs and be more clever and smarter with how you're developing drugs to treat different aspects of life with that. And actually, I think you have a company just in that region.
So I didn't, obviously you can ask me, why is it interesting to know about proteins? But also, almost all the drugs that you take, not all of them, they are either activators or inhibitors that are directed at one particular drug target, which is usually a protein. And in the human genome, for example, putting aside pathogenic organisms, but there's, let's say 20,000 proteins. So you could argue that a complete pharmacopoeia of drugs would mean you'd have one drug that inhibited every one of the 20,000 proteins in the human in one set of test tubes, and then another drug that activated them. And usually in disease processes, to cure you of, say, cancer, usually there are half a dozen genes, or some cancers just 2 or 3, others 20. You might want to either block or activate more than one gene. And there aren't, there are probably drugs against several hundred drug targets, but there is not yet a complete library of activators and inhibitors for all the protein targets. And of course, there are other targets like viruses and bacteria, which you want to get rid of. So for example, in coronavirus, there are 2 drug targets that people think if we had really good drugs, which we do not have yet, that would block the activity of the protease that cleaves up the polyprotein that's absolutely essential for coronavirus. And there's the RNA polymerase that copies the genome of virus. These are the 2 prime drug targets, and there are hundreds of companies working on them. And they all have now in the lab really good in vitro inhibitors that block the activities of both of these drugs. But it will take 3 or 4 years until they feed their way through to being approved for use. And then, if it all goes well, you'll take a pill or 2 pills or 1 pill that has both drugs in the same, and then it will completely stop coronavirus dead. You won't even need to bother with vaccination, but that's some years off. And in HIV, that is the current situation. There's, a multi-drug treatment that will stop HIV in its tracks, and people who were dying 6 months after getting HIV from AIDS, now they can live forever, but they have to take the drugs, indefinitely. But with coronavirus, you probably only have to take them long enough to clear you of the virus, and then you can stop taking them again. Yep. So it probably needs some investment, and the drug companies don't like, in terms of a business model, a drug that you take one of and completely cures you forever is not a very good business model. What you want is a drug that when you get the disease, and HIV is such a one, you have to take it every day for the rest of your life. And so, drugs against Alzheimer's disease, they've got a lot of investment, but drugs against coronavirus that give you it, they will need to have some government or state or World Health Organization support to create the impetus to develop them. But the companies are all ready and waiting to take the money. So I just had lunch today with Malcolm Weir, is the CEO of the company that we started about 14 years ago. And this is Heptares, Sosai Heptares. And they've got about something like 40 drug targets. But only 1 or 2 of them have actually reached the stage of being in humans in clinical trials. not yet so it takes, for small molecule drugs or even for antibody drugs, if you're putting them into people, it takes many years of clinical trials. And the great thing about the vaccines, they've been able to do a very fast track development of it. And now in Britain, at least, in the US and some other countries, the vaccines have worked, but it'd be much better to have drugs. And these are inhibitors, in this case, against proteins. In the case of coronavirus, the spike protein that's on the outside of the virus, and the polymerase that makes the next virus once it's infected the cells. Those are the two prime targets, and people who do proteins and drug companies that develop drugs against the proteins, they are the people or the experts who can do the development, but they need to be pushed and funded by investments either from themselves or from venture capital or from governments or from the tax base. So, but it's working. I mean, let's say before 1940, there was very little. I mean, penicillin's probably the first one. That was just pre-war and during the war. And then all the antibiotics, and now it's a big industry, actually. Pharmaceutical industry didn't really exist 100 years ago.
So just rewind the clock back. You've got a spin-out company. Well, it's an established company that you spun out or started. So that's another complete dimension to your career. And you start. So why as a physicist did you choose to then go into biology? You're not the first. Actually, I think Dan Davis was exactly the same. Did his degree in physics and then went on to become, a star of immunology. Why biology? What attracted you to biology?
All right. Well, so I can, I've told this a few times actually, but when I was in school, I really did enjoy maths and science, and I couldn't wait to get rid of the burden of having to study other things like English, French, Latin, whatever. And so when I was about 13 in high school, we were asked which subjects we would like to select for secondary education. It was the Highers, which in Scotland is like A-levels in England, or baccalaureate or whatever. So I filled in my form and I said I was going to do maths, science, and technical subjects, engineering.
Yep.
And then at this point, English was compulsory, but none of the others. I'm called into the headmaster's office, aged 13, and they say, you know that you won't be able to go to university unless you have a language, a foreign language. And so I said, I'm only 13 and from the rural parts. I hadn't heard of university at that point. We, our family, we universities didn't exist. We live in the countryside. So I said, okay, well, that seems like good advice. So then I look at my ability in languages, and I had studied aged 11, 12, 13. I had studied French and Latin. And in the exam, just before I decided to give them both up, my score was 47% in Latin and 48% in French. And 50% is supposedly the pass mark.
Okay.
So I said, okay, 47 versus 48. I better do the 40. So I chose French.
By 1%.
So yeah, 48% in this is the end of 3 years of secondary. So then I studied French for 2 more years. And I sat my lower Scottish exams, which is equivalent of O-levels or GCSE.
Yeah,
now.
so you're 16 years of age.
Yeah, and so I'd be 15 or something like that, taking my O-levels, and the only one I failed was French. So then, and I was the only one in the class who failed because I was obviously the most marginal, and I was only taking it because the headmaster told me I had to. So then in the 6th year, which is the final year of school in Scotland, I'm in the class and is the teacher, and there are only 2 of us left. Me, because I had failed lower French, and another young girl, same age as me, but she'd failed higher French because she'd picked. So
Yes.
we had individual tuition for a whole year, both of us, one teacher, two pupils. And at the end of that year of essentially, several lessons every week for the whole year, I did pass. I got by then they changed the result, and the grading was A, B, and C. So I scraped in with a C, and consequently, I was allowed to apply to university. And so, but at that point, I finally could give up. And then I did physics and maths in Edinburgh. And then, of course, the question you're asking is, how did I get from physics and maths into biology?
Why choose it?
Yeah. All right. So I always studied carefully each step in my career. And in my, final year of physics, so I gave up maths after 2 years, because you had to, I did physics and maths for 2 years, and then you have to choose. So I chose physics and gave up the maths. So then the final 2 years, 4-year degree in Scotland, 3rd year and 4th year was physics. And at the end of that, I thought, well, what should I do next? After high school, scraping through with the French, physics degree, doing well in physics, I have to say, because I enjoyed it. I thought I'd better go and see the Careers Advisory Office in Edinburgh University. So I went along to the Career Advisory Office, and they said, oh, look at all these jobs you've done in the summer to earn money and physics. And they looked at the grades, and they said, it's clear that you should do a PhD. So I hadn't really heard about PhDs either, but nevertheless, I thought, okay, I better look into this. So then the final exam came in physics at the end of the 4th year. And in Edinburgh, it was 6 papers. So 5 of them were on physics, optics, electronics, and so on. But the 6th paper was always considered to be a bit of fun. You had to write an essay, and you were supposed to be somewhat philosophical. So my essay at the end of my physics degree, it was multiple choice. They were just giving you the opportunity. Yeah. So one of the topics was, would you like to write an essay for 2 or 3 hours? This was and I picked writing an essay on time. Now, time normally means relativistic time and so on and so on. But I decided this was a fun thing, and because I'd been thinking about it a lot, I wrote that time consists of the past, present, and future, but since we know everything about the past and the present, or at least you could look it up, the only thing that's interesting about time is the future. And so then my little essay was about what is the future of physics. So then the rest of my several hours of essay consisted of a little paragraph about astrophysics, which would be cosmology, gravitational waves, so on. A little bit about solid state physics, which would be transistors and computers and so on. A little bit about single particle physics, CERN and particle colliders. A little bit about fusion research and how it's going to produce unlimited power. And then the final one out of about 5 or 6 areas was biophysics. And I said, okay, biophysics needs physicists to do physics in biology. And we had a very good physics teacher at high school called Bill Cow, who was just an enthusiast. And he was keen also on biology. And he had recorded a lecture by one of the lecturers in Edinburgh University called Jack Dainty. And he was in the Edinburgh University biophysics department, but they would never make him into a professor. He was a lecturer or a reader. So eventually, and he was there still while I was at university. I never met him, but our physics teacher recorded Dainty giving a lecture while we were still in high school and played it back to us. And it was Jack Dainty. He was putting an electrode down the middle of Nutella, which is a plant cell with a big cell, and looking at electrical currents in
Yeah.
Nutella. And Bill said, this is the future of physics in biophysics. So 4 years later, I went through all the different options and decided that he was right. And so then I said, okay, now I've decided on biophysics. Where in Britain can I go? I didn't want to go out of the country. So I then tried to research biophysics all over Britain. And in the physics department, the Medical Research Council, where I ended up, nobody had heard of it in the physics department. I hadn't heard of the Medical Research Council. But in our physics department, there was everybody who was doing a PhD or thinking of it. There was the Science Research Council in those days, which is now metamorphosed into 6 different research councils. So the SRC had a handbook this thick, and I looked through it, found biophysics, found all the biophysics departments in Britain, which was, Oxford, London, Leeds, Norwich, and Dainty by then was in Norwich in my final year. So I wrote to them all and said, what do you do PhDs in biophysics? And several of them, Leeds and Norwich, wrote back and said, we don't allow anybody into our university to do a PhD. You have to do an MSc first. And I said, well, I've had it with exams, no more exams for me. So I went round anyway, and I went to King's College in London, and it was John Randall, Maurice Wilkins, Jack Lowy, Jean Hansen. It was a really good department. So I went there in, November 1965, all the way down to London in our old car. Went there for a day, and I thought it was quite a good department. So I wrote to them and said, I've decided I'd like to do a PhD in biophysics at King's College. And they wrote back to me, Strother Arnott, saying that, okay, thank you for your application, we'll let you know. So they didn't say I could come. So I thought, well, maybe, while I'm waiting for them to reply, I should go and tell the head of department in Edinburgh, the physics department, who is called Bill Cochrane. So I made an appointment, went to talk to the head of department, said, I am in your final year physics class, and there were only 40 of us, so he probably knew who I was. I said, I've decided to go into biophysics, and I'm going to King's College London. And he said, no, he said, you mustn't do that. So finally, I found somebody who knew about it.
Yeah.
I'd never talked. He said, you must write to Perutz in Cambridge. So I wrote to Perutz, January 1966, and heard nothing back. So after about 6 weeks, I'm wandering around the physics department in Edinburgh saying, this lab in Cambridge is no good. I wrote to them 6 weeks ago. They haven't replied yet. And they said, I said, I'm thinking of writing another letter asking them whether they, and they said, oh, no, don't do that. They've been writing for references. So finally, I did get a little note from Max. One line said, Dear Mr. Henderson, thank you for your interest. Would you like to come and visit the lab? This is where I am now in the building here. So I immediately wrote back saying, Dear Dr. Perutz, yes, I would like to come and visit the lab. Could I come next week? So then he writes back immediately. He said, yes, you could come next week, but it'd be much better if you came 3 weeks on Saturday when we have a special open day for students.
Yeah.
So I went on the train overnight, no hotels, no staying. So I get an overnight train to Cambridge, arrived at 8 a.m. at the station on a Saturday morning, came to the lab, and there were about 30 students, 28 of them from Cambridge, and one from London, one from Edinburgh. And they're all working. Saturday morning, they're all working. I thought, this is better than King's, because on Wednesday, 5 o'clock on a Wednesday afternoon, it was getting pretty quiet, and I was about the last one to leave. But in Cambridge on Saturday morning, they're all working. So I said immediately, So I withdrew my application to King's and applied to the lab, and they accepted me about 2 days later, say, doesn't matter what your degree is, you can come. So they didn't even care whether I had a first, second,
Yep.
So anyway, I ended up here, and I've been here for 53 years or something like that. So there you go. That's how I got into biophysics.
That's a long, long time. There's two things I'd like to pick up. The first one, it's more. I'm just more interested. What was your car that you drove down to London in?
Had I had time to show you, I have a beautiful photograph of the car. Actually, I can send it to you. It was a Morris 8 Series E, 1948, and we bought it in 1962 when I was 17. So I had two friends who were also studying physics and also came from the same class in Boroughmuir School in Edinburgh. And when two of us were too young to drive, so one of the three of us had been taught by his father how to drive. So the three of us, we each put £10 into the kitty, and we bought our car for £30, which was about 10% of the new price.
Yeah.
And it was a 14-year-old Morris 8 Series E, and it worked actually. It went wrong a lot, so we became very good motor car repair engineers. But the one friend who could drive taught the other two of us how to drive, because that was our hobby. And of course, we both passed our test within 8 weeks or something like that. And then we had one car between three of us, and we would just share it. So we would take it to the Highlands. I took it around Ireland with a couple of other friends once, and it broke down a lot, but we got expert at fixing it in the end. And then after about a year and a half, with the three of us owning it, one of the friends decided he wanted out.
Yep.
So we each gave him £5 to buy out his
Bought
share.
his share.
And then about a year later, my parents moved out of Edinburgh into the country, and I needed transport. So I traded in my share as well for £5 or something like that, and I bought a Vespa motor scooter. And because, we didn't have much money, but we lived in Edinburgh, but we were about to move to Broxburn, which is about 15 miles from Edinburgh, in the countryside, sort of. But I'd noticed that in the backyard of our house where we lived, right in the middle of Edinburgh, there was an old Vespa that had been there for 6 months. And so I asked my mum, who was quite well organized, could she find out what that Vespa was doing? And she found out that it was owned by somebody who had bought it. They were new then. They were probably about £100. So this guy, who was in his 20s, had bought a Vespa scooter for about £30 and didn't like it. So he tried to sell it back to the dealer, and the dealer said, okay, he said, I'll give you £10 for it. He said, that's rubbish. He said, I could make more money by breaking it up for parts. And so he wasn't knowledgeable. He cut out all the wires, everything was just complete mess. And so it didn't work. So I bought it for £7. So I sold my car, my share in the car, my half share, which would be £15 initially, sold it for £5 and bought the scooter for £7. And then reconnected all the wires and got it all working. It never was quite, but it worked. And then it turns out riding a motorbike or a scooter in Scotland in the winter.
It's not very clever,
is it? It's a dangerous thing, actually. And one morning, I was working on the post at Christmas to earn money, delivering the post. It involved driving from Broxburn into Edinburgh at 7:30 in the morning on my little Vespa scooter. So I'm going along 45 miles an hour. And, it's very early morning, not a lot of traffic. and I had this feeling as though I was floating on air. And it turned out it was black ice, completely invisible, and friction coefficient of 0. So eventually the wheels have lost traction and the scooter swung to the side and then went back, and eventually I fell off and I skidded along for 20, 30 yards, but it's completely smooth. So my trousers are undamaged because I'm on ice.
You haven't stopped yet, though.
Pardon?
You're still sliding at this point. You haven't stopped. It's the stopping that hurts.
Yeah. Scooter is sliding. It stopped before me because it had metal. I slid on, but I got up. I mean, it's in a big motorway road. Fortunately, there were no cars coming. So I got the scooter up, got back on, and then was a bit more careful because I realized now it's icy. But at that point, I decided that perhaps it was time to trade in the motorbike scooter. So then I did buy, having saved up some money, I did buy an Austin A30 for £60. And then I had that for about 5 or 6 years. And then when we went to America to be postdocs, I eventually sold my £60 Austin A30 that I'd bought, that I'd used for it. I sold it to the same friend who bought my share in the Morris 8, and I sold it to him for £5.
He seems to do quite well with £5, doesn't he?
Exactly. So you can see that I had a lot of knowledge and expertise, but no money. So it's amazing what you could do with that would be about £50. So you'd be selling a pile of old junk for £50. But you can easily buy a car now. You can get beautiful cars for £50 or £100, and as long as you're willing to fix it yourself. If you want a garage to fix it, you're better to spend a bit more.
So there's two things. Don't take this the wrong way, but you're a very clever man, or so I thought, until you bought a scooter, a Vespa up in Scotland, because that was always going to happen. That was always going to happen up there. So the other side is you said that was your hobby, was your car and fixing your car, engineering your car. What other hobbies do you have? Because you've gone down. You're at the LMB. You're working 6 days a week. You're working on Saturdays. What did you do outside of work?
At what age?
Go on.
When I was a young lad, I didn't have any hobbies at all, actually. I have so many hundreds of stories, actually, but it's because they impressed it on me. But I was brought up in rural Roxburghshire, which is 2 or 3 miles north of the border with England, and my mum tried to get me to read books, and I did try, but I never managed to read a single book of any kind. And I had my mother's family lived in Edinburgh, and we visited once a year for a week for a holiday. And my grandmother's sister, so my great aunt, she owned a little corner shop in Edinburgh, in William Street, and she sold newspapers, firewood, firelighters, cigarettes, chocolates. It was a general store. It was like Sainsbury's, but tiny. And she had to work from 7 a.m. till 7 p.m. every day. And she didn't have any children of her own. But the great thing for me is that every week from the age of about 6, she sent me every single child's comic in the newsagent's shop. So every week we got this package, which came from the grandparents and the great aunt. And it would have things for my parents in it. But for me, it had maybe 15 comics. Everyone, the Lion, the Dandy, the Beano.
What was your favourite?
Pardon?
What was your favourite comic?
The favourite one? Good question. I think there was one called they didn't have the one I would have liked, which was not in my aunt's shop, was the eagle.
Okay, yep.
So
I've heard of it, at least.
But I'm leading into it. So that's what I had. And then since once I'd read them, they had no further value to me. So I'd give them away. And as a result, I became quite popular for giving away free copies of 1 week old comics. But in our village, there must have been 800 people. The school had about 150 pupils in it. Our class had 20 pupils, 7 classes, 150 people. The year ahead of me, one of the people I knew was another young lad, a year older than me, and he was the son of the headmaster of the school. And I was slightly friendly with him. But one time when I'm about 11 or 12, something like that, he went to the same secondary school as well, and we went on the train every day from the village where I grew in Newcastleton to Hawick, which is 20 miles north. So every day, morning and evening, we went on the train, and of course there were 20 or 30 other children. And the headmaster's son went on the same train, so I was slightly friendly with him. And one time I'm visiting his house, and I notice that he has a copy of a little magazine called Boy's Own Paper. Now, you may not have heard of this, but that was a very boy-oriented paper, with adventure stories and so on and so on. And so when I got back home, I told my mum, I said, there is this very good magazine, which my aunt doesn't send us, because she didn't have that. She didn't have the Eagle, and she didn't have the Boy's Own Paper. The ones she did have, though, were things like The Wizard. There were a lot of child's comics. Anyway, I persuaded my mum that she should buy me once a month the Boy's Own Paper, which is like £1 or something like that.
Yeah.
And in a way, that was the first time I became any interest. So one of the issues, which is, January 1959, which is about, I don't know, 13 and a half. This was the issue. It came, and it had a center page pull-out spread. It said, build your own 1-valve shortwave radio set. So I read this. I thought, this is really interesting. We're cut off here in the middle of nowhere. We have no contact. We did have a TV that worked very badly. Reception was very bad. There's no transmitter for 40 miles. So I decided my project, which is the beginning of the first hobby, I would build this 1-valve crystal set. But we had no components whatsoever. So I wrote to my grandmother, who lived in Edinburgh. I said, I need the following components. And in Edinburgh, they had lots of shops, of course. So my grandmother would go to an electronic component shop called Brown's on the Mound, which isn't there anymore. And she sent me the valve, the tuning, the coils. And there were only about 8 components. And I wired them all up according to the center pull-out spread, and it didn't work. So, I spent months making this thing. It didn't work. So then I had to compare the physical layout with the schematic wiring diagram, which, of course, I'd never looked at before. Yep. Then I realized that the coil, which was the official one I was supposed to use, they changed the design between when they did the article and when I got my version of it. So the connections were wrong on the coil. So when I connected them correctly, it worked perfectly. So then I had a 1-valve radio that I could then listen to myself without having to bother anybody else. The only thing was, it was you don't get much gain out of 1 valve, so you have to run it close to being a transmitter. You're almost oscillating. It's called a regenerative receiver. And as it crossed over from being an amplifier to a transmitter, it would broadcast a lot of interference. And our neighbor, who was the next house, complained that she'd been getting a lot of interference on her radio reception. And that was my shortwave radio.
It's all your fault.
Being a transmitter instead of, anyway, and that was partly, and then from then on, I became interested in electronics and, and actually, unfortunately, when we went to high school on the train, we couldn't stay. So I couldn't go to, they had after school clubs, but because there was only one train back, I couldn't go to any of them. So it was all self-taught hobby. And I did that probably till the age of about 20, something like that. And then when I left home to come to Cambridge, I left all my electronics and, power supplies and stuff in my mum's house. And after about a year and a half, she didn't tell me, she decided to get rid of it.
She threw your hobby out, quite literally threw your hobby out.
Well, she gave it away to another teenage boy. So I don't have any of those things anymore. But the ones that I'd kept and taken with me to Cambridge, when we eventually went to America, I had to get rid of those as well. And so I sold some of them to another friend that had been from school and university. And unfortunately, he died age 60. So he'd had this stuff that he bought from me for 40 years. So I got them back. So now I have in our house, I have a 1942 working shortwave radio that is 1942, that's 78 years old. And because we were locked up with coronavirus for the last year, I decided that this was the perfect time to fix all the old clocks, radios, and broken stuff around the house. And when I switched it on about 6 months ago, it didn't work, of course, because it hadn't been switched on for. So I did spend about a week replacing all of the electrolytic capacitors, which of course, they dried out and so on. So now it works perfectly again. It's almost as good as the ones you buy that have got microchips running them.
You fixed it, but it sounds like it's going to be a new one at this point. just kind of replacing all new parts. It's kind
well, no. I left all the old components are there. So, with electrolytics, you can just wire a new one and they're much smaller. You just wire them in parallel and then you just ignore the old one. So I mean, it works quite well, actually. It's really good.
So I've now got to ask this question. Are you a minimalist or are you a maximist at home? Because it sounds like you hoard a lot of stuff.
Yeah, we never throw out anything. Yeah,
You're a hoarder. Well,
no, I've got numerous others. You have to bear with me, Peter. We have a reasonably big house, but when we moved into it 22 years ago, I thought, since it's empty, this is the time. And so we put floorboards in the attic. So our attic is full of pre-war radios, TV sets, record players, and then all the old papers and files, meetings and things, all up in the attic. And that idea that you can use the attic came originally from the house we lived in Broxburn, which was, the second 2 years of my 4-year degree in Edinburgh. And my parents moved out to a less expensive house in the countryside. And it was a terraced block with about 10 houses, but each of them had an up and a door. So it was 20 houses. I don't think it was very expensive. It had one room that was pretty small, which was the kitchen and the sitting room in one. And then it had 2 bedrooms. My parents were in one bedroom, and my brother and I were in a bunk bed in the other bedroom. And that was our house. And then when I'm about 17 or 18 and in university, I'd noticed that there was a hole up into the attic, not very big. So we looked up there, and then when you go up into the attic in these old, you can look along, and it was the attic for the entire block of houses, and there's nobody up there. So I asked my mum and dad, I said, do you think we could put a bigger trapdoor? So we bought a loft ladder.
Yep.
And then you could get up into the, and then we got a little skylight put in, which was, very cheap, just a pane of glass, you could open and close it. And then this friend and I, the one of the people who shared the car, his father had a bigger car. It was an estate car, so it opened the back. One day we drove down to the docks in Edinburgh, where they had a shipyard and a wood yard and so on. And we bought for £8, we bought an entire carload of floorboards, and then took them back. And then I spent probably about a week flooring the loft completely, the entire house.
Just above the bit above your terrace?
Yeah. It was a bit about our terrace, but actually, you could go a little bit further into the neighbours.
A bit of space.
There was nobody else there. So then we had a loft ladder, a skylight, and a floor, but no walls. And so we put 2 single beds up there. So one for me, and then one means I could have a friend to visit. And then my brother could have the proper bedroom. He had that to himself, so he was quite pleased. This is about like 6 months after we moved there. And then I probably, I was only there for 2 years. But during that time, once it became clear it worked quite well, I added walls.
That's good to know.
4 walls, but I left it so that you could remove a bit out of the wall and put stuff behind the walls
in the other people's attics.
Yeah. Well, the other people's. And then we've continued to do that. So in our house, we then, when we came back from America, when we were, let's say, late 20s, and we've got children now, we did the same thing in the semi, and it was before they had. Now they don't do this. They have dividing walls in the attic, so you can't go in. But we went up into the semi-detached house, and it's the same, our side and the neighbor's side. And so we floored that as well. And then so from teenage we've always had homemade floored attics, and they're always the complete size of the whole house. So it's an immensely. So I recommend anybody who lives in a house with an attic that they should floor it and get a pull down ladder, and then it's not only another room; it's another room bigger than any other room anywhere in the house. So,
see, my father-in-law has his train track at Model Village up in part of his loft, which is when you take the grandchildren down, they love it. Actually, I think we all quite like it. It's really cool. Jerry's
still there, is it?
Yes, yeah. So again, he did just part of it. He's got a part for storage, and then he's got this lovely bit. He's done an amazing job. So I guess his hobby at the time is setting that up and still running. So that's brilliant. And actually, I say grandchildren, you'll have great grandchild, not from us, not from my side, that will be enjoying that very, very soon. Some other questions, Richard, because I realise time is flying past us really fast. Can I ask you some quick fire questions? Do you prefer email or telephone?
Well, I'm happy either, but hardly anybody uses telephone nowadays, actually. So I have possibly, I don't know, 50 or 100 emails a day and no phone calls. And even the children now and the grandchildren, they do FaceTime or Zoom or Skype.
I should have probably said email or Zoom or equivalent. It's probably
I do mainly. I've specifically never joined Twitter or Facebook. And actually, I remember, Brian Hartley, who died very recently, 95. He was one of the mentors in the early LMB building. He said, I never read the literature. He said, if something important happens, somebody will tell me about it. And so with Twitter, normally in a normal day in the lab, I'd be here working, and then I'd go for coffee in the canteen. And to do that, normally we walk through, there's a shared terminal room where people can go with slightly more powerful computer. So one day I'm walking through the terminal room, and one of the scientists there, Paul Emsley, said, "Oh, have you seen Twitter today?" I said, "No, I don't read." So he was telling me what he'd read in Twitter 5 minutes before. So, it was really hot off the presses.
Yeah.
And so there are, I would say most people do have a Twitter connection now, 90% of the younger generation. And so they tell you what's happening. But, I just don't have the time to do it. You can't do everything. You have to be careful and focused with your time. And one of the great things, unfortunately, the bad thing about the coronavirus is your normal social connections, going to meetings, so on and so on. But the great thing is no traveling, and you can get a lot more work done. And if you don't do Twitter and Facebook, that must save you hours a day.
Do you know what I think? Without the travel, it's just made life busier because you haven't got the downtime when you're sitting on a plane. It's kind of the one time you could get away from everything in a way. So you said you never read a book as a child. What do you prefer to do now? Watch TV or read a book?
Well, I tend to, I do both of these things actually. But with books, I tend to, anything I think I need to read, I buy it, and I don't throw them away. So we have increasing, and so often you can skim a book, in a few minutes, randomly read, you get the whole idea of it. So I have quite a lot of partially read books, and then I do watch TV, and particularly over the last year, I am now very well versed in films from the 50s and 60s.
What do you watch though?
What do I watch?
These 50s, 60s films.
Well, there's a lot of Hitchcock. All the Hitchcock films from 1935 onwards, they're very, very, very good. But let's say of the things I've watched recently, for something like this, bite-size, whatever. The one I can strongly recommend, and I thought was wonderful, was the series called Breaking Bad. Now, you must have watched this.
I haven't watched it, but I do know of it, of course.
Peter, you must watch it. And I'll tell you my, two little mini things about. I had not heard of it because you wait for things to arrive. But my son, who's now in his 40s, he said, "Dad, you must watch Breaking Bad." And I said, "Oh, well, I will." But then I hadn't watched it. So next time I visit him, he says, "Let's watch the pilot opening episode, which is a 1 hour, all the others are 45 minutes. And so I can recommend to you, Peter, watch the Breaking Bad pilot episode, 1 hour. And then the second tip, so Alistair and his friends had found out about this 5 or 6 years ago. And they had bought a boxed set for £40, and they passed it around and they watched it. But by the time he had found it, it was on Netflix. And this is the thing that made Netflix take off. My son, Alistair, said, if you subscribe to Netflix, it's free for the first month. And you can watch Breaking Bad for nothing. So I signed on to Netflix, and it's 64 episodes in the complete series. So it's probably about 45 hours. And I watched, maybe two a night, sometimes 3, sometimes none. But I managed to watch the entire series in 3 weeks.
Did you unsubscribe from Netflix at the end?
Yeah. And then I said, Netflix is so good, I'm going to subscribe.
Okay.
And now the children, the grandchildren, they all subscribe to Netflix. And it is still very, very good. A lot of other companies now are catching up on it. So Breaking Bad, it's about, you must have heard a story.
We'll move quickly. So I will watch it. It's
a 52-year-old chemist, a chemistry teacher in high school. He's the main character, Walter White.
I will. I promise. I will watch it. I promise that.
You'll find it if you have it has a scientific underpinning, and it's terribly well written. Actually, you'll really enjoy it. Anyway,
so I will give it a go. So the next question is: do you have a pet hate?
Hate?
Yeah, a pet hate. Something you just really you don't like, winds you up. Just what niggles you?
I don't. I don't have any hates, but I have a lot of things that I find very amusing. And so things that I find very amusing would fall into the category of things that other people hate. For example, I find it very funny how easy it is that some people can delude themselves into believing something. And for example, in science, they have some theory about biology or chemistry. And to any normal person, you can tell it's complete rubbish. But somehow or other, the world has come in and they're focused on it. And they are completely... So the two interpretations of somebody who has published a wrong paper or has a wrong idea, the two possible explanations are, assuming that they're wrong, either they genuinely believe it and they're deluded, or they're dishonest and are doing scientific fraud.
Yeah, that latter one's really bad.
So, you have to look at a particular thing and you have to ask yourself, is this a naive, deluded person, or is this a devious scientific fraud person? So I find this very amusing. But not every people think. You think, oh, you're being wicked, but no, I think it's just funny actually.
Okay, so I'm going to challenge you now. Do you think people thought you might have been a fraud when you told everyone that X-ray was no longer the way forward and they should all be going towards electron microscopy? Because there must have been a load of people there thinking, wait a minute, this is not right.
Well,
you were proved right. So you're being proved correct, but...
Yeah, no, but you have to realize, I came from fairly non-academic rural background. So for me, every step is a step forwards and upwards. So I've never, ever had any doubts about this thing. But I can tell you one interesting story is actually, there are many related to this. But the reason that I said that, I wasn't looking to do it, but what happened was we all assumed that everybody knew that radiation, X-rays or electrons did radiation damage. And from about 1980 or 1990 onwards, it was clear what it was. And I thought everybody just knew this. The first thing was when Ada Yonath, who, has a Nobel Prize for the ribosome work, she was one of the first people with Huck and Hope to freeze crystals and put them in a synchrotron. And she said at a Gordon conference in, New Hampshire, she said, when you freeze a crystal to liquid nitrogen temperature, the diffraction lasts forever. There is no damage. And of course, we'd been doing electron microscopy for years. We knew when you put the beam on it, it lasts for a few seconds. So we said, that must be wrong. And so one of the first things I compared in 1990 X-ray and EM, and showed that in order to find X-ray damage in 1990, you'd need 3 days irradiating in synchrotrons then to find the damage, or 5 years on an X-ray source, and just nobody had done it. So that was a paper that was published in 1990. I assumed everybody knew that. But the people who are running the European synchrotron facility that was being built in Grenoble were trying to build different beamlines, and they had a meeting to discuss X-ray microscopy. And it was Carl Brandin and Andrew Miller, the director and designate director and acting director and so on. They said, would I come along to the meeting and give a talk about radiation damage in X-rays? So I thought, well, fine, this is common knowledge. So I went along to this meeting, and there are 50 X-ray microscopists who think X-rays is like best thing since sliced bread. And I stand up and I explain that electrons do 1,200 times less damage than X-rays. And they were really angry because they wanted 5 million euros to build a beamline. And I'm telling them that it's rubbish. And I almost got lynched, actually. And so that was the reason I decided I better write if these 50 intelligent people, the best in the world, X-ray, didn't know this, then somebody needs to raise the profile. So I started to write a review. It was comparing X-rays and electrons. But during that time, a journal called Neutron News, it's still published 3 times or 4 times a year, came with all the cross sections for neutrons. So in the end, I wrote this article, neutrons, electrons, and X-rays, which ones are the ones you should do in theory. And it turns out not only are electrons 1,000 times less damaging than X-rays per useful scattered event, they're 3 times less damaging than neutrons, which are thought to have no damage. And so I wrote that review, and then I was, having written it, I was then personally so convinced that we stopped all our work and then decided to focus on single particle electron cryo microscopy. And Howard Hughes once had a meeting. I went along, and Michael Rossman, who was here in the lab when he was a postdoc, with Max, went to Purdue, died about a year ago. I was asked to give a talk at Howard Hughes, and Michael was in the audience as listening to this. And after I'd given my talk saying 1,004, Michael stood up and said, well, he said, this is not what I've been teaching my students, for the last 30 years. He said, must be wrong. And then, but what he hadn't realized is that it is true that electrons are actually 100,000 times more damaging in terms of their cross-section. But if you look at the ratio of scattering to damage, it's 1,000 times worse than X-rays. So it was just a learning experience. And so everybody then accepted it, and then we went on. So no, I never had any trouble convincing people, because all you have to do is write down the facts. And eventually there's this review, which is now, it wasn't cited much at the beginning. So it's a paper that we call, when you publish a paper, you may have seen this yourself, you publish it in a journal, and the journal has an impact factor, number of citations on average in the following 2 years, and it has a half-life, because normally when you publish in Nature, it gets cited for the next year, and then it decays the second year, and the half-life is usually about 1 to 2 years. But if you have a really good paper that's what they call seminal, it has no citations in the first year, and then it just grows linearly with time. So actually it has a negative half-life. So we think that the best papers have a negative half-life, and they have their own momentum, and they grow. And
so that's because it's also because others aren't doing it, and if it's a technology, there's not many people can cite a new technology until they've done something with it, and then it will grow. And obviously, massively in the case of structural and cryo EM itself.
But DNA sequencing, cloning, all those papers, they'll be in that. They're all in that category because they were new ideas that then everybody cites until they get tired and then they cite a textbook or something.
So I cannot believe we've been talking for over an hour, and these are meant to be an hour long, and I've got so many questions I still want to ask you. So do you know what? I hope no one minds if you just go on just a little bit longer.
I'm happy to do. I can do 10-second answers as well. I just wasn't clear.
No. Do you know what? What's your favourite music?
Good question. I wasn't a very big music fan, but I did decide I needed to it. So Bach's cello concertos.
Okay. What's your favourite food?
Well, I had sausage and mash for lunch today.
With onion gravy?
Yeah.
Of course.
It was Lincolnshire sausages with, it was really good.
Okay.
And also, when I was young, we had at the bottom of our street in Tweedmouth, which is the opposite shore of the Tweed from Berwick, fish and chips is very good.
Oh, yes.
Probably bad for you, but anyway.
In the right place, at the right time, in the right place. What about, this is an odd question. What is your favourite item of clothing?
Good question. Well, I have never changed my tastes, and I've worn sports shirts and sweaters for my entire life.
That's a perfect answer.
I've never had a beard, except for 2 weeks when the children wanted me to grow one. Then I cut it off. So I'm afraid I don't really change. I'll tell you one thing that might be interesting. When we were children, we were always asked, what is your favorite color and what is your favorite number? And I always said it was purple and 7. And later on, when I was about 25, I started to work on purple membrane.
Yeah, of course, with a 7 plus protein.
A couple of years later, we discovered it had 7 transmembrane helices.
Yeah.
I thought it was pretty good.
It was fate. It was fate.
Yeah. So,
I was going to mention. I know you like hill walking. Do you still kayak?
Yeah, we have 2 in the garage. Don't go so often now, though. But
it's amazing.
Actually, the same friend who had the part ownership of the car, last time he visited Cambridge, we kayaked from one of the villages into Cambridge. You always paddle down. The key thing about kayaking, you always go downstream. And then even if you get tired, you just wait and the river takes you to where you get.
I thought you were going to say that your friend came down and bought it off you for a £5. It's amazing because actually so many scientists that I've talked to for the Microscopists kayak. Jennifer Lippincott-Schwartz still kayaks to work over to Genelia Research Campus. Alison North still kayaks on her holidays. It's amazing just the number of kayakers.
I bet you Jennifer Lippincott-Schwartz doesn't kayak in both directions. So she probably lives upstream and kayaks in the morning and then takes it back on her car roof rack.
I think she goes both. I don't know. I think she might go both ways.
It's difficult. Well, unless it's there is a river right there.
She's going across. So it's the River Selden, and she's going across. Or Selden Island, isn't it? So it's
right
over there. So yeah, so that's quite entertaining listening to about when it gets too dark is maybe not the best thing. But there was a few with that.
Anyway, all our best kayaks have been taken by the children, so we only have the ones that are not terribly exciting left.
I do have. Maybe we'll do another one of these and come back to more questions in the future because this has been so brilliant. I'm going to ask you though, what do you think is the biggest challenge going forward, scientifically? What's the big science challenge that needs to be addressed?
A lot of the young students and postdocs ask that question, actually. So the biggest challenge going forward, or at least the one you might want to work on, depends on how old you are. Okay, so if you're 75, this is not the time to start on a project that's going to take 30 years. But when I was a first-year postdoc at Yale, I thought I should simply, having worked on an enzyme for my PhD, I thought I'll just pick another enzyme slightly more interesting than the first one and work on that. And then there'd be plenty more. But when I told my postdoctoral sponsor this, he was a very clever Chinese professor in the chemistry department. Actually, chemistry department at Yale is where I did a postdoc, without any background whatsoever in chemistry. We stopped with inorganic, aged 14 in high school. But he said, no, he said, I don't think you should work on an enzyme. He said, there are thousands of enzymes in the world. When you're 25 years old, you should pick a topic to work on that will come to fruition in 20 years. And so I said, this is very good advice. So I stopped working on enzymes and started to work on membrane proteins,
Yep.
which in 1970 was very, very badly understood. So now, I mean, I think the big frontier is the neurosciences and how memory and learning works in the brain. But I would say there isn't really a big rush to get into that just at the moment, because the techniques that exist at the moment are not powerful enough to get to the bottom of it. You can make progress and eventually there'll be a breakthrough, but what you really need is 1 micron and 1 millisecond resolution on the entire human brain. And of course you've got, 15 centimeters, so that's 150 millimeters, 150,000, that's nearly 1 million pixels in each direction. So that's 10 to the 18th pixels. And if you were to record every millisecond activity, you'd be talking about 10 to the 21 numbers per second. And this is beyond any capability at the moment. And so you have to, dissect it down into more digestible lumps. And that's why a lot of people are working on Drosophila brain. There's only 300,000 cells, or the nematode brain, only 300. But the human brain, it'll never be the last frontier, because you can always invent things. But understanding learning and memory and how we think and work, that is. And I remember having a chat with Colin Blakemore, who's a neuroscientist who worked on cat vision and was the head of the MRC for a while. And I sat beside him in a lecture. I said, this neurobiology is very interesting and obviously many things to learn, but how long do you think it'll be before we understand learning and memory? And after a lot of hooing and hawing, we both decided it's at least 30 years in the future. So, there's no hurry. If you spend 5 years doing something else and you're a young scientist, you can easily get into it 5 years from now. There's no rush. In fact, it might be better, because one of our favorite things is, if you're wrestling with some technology thing, one solution to solving the problem, instead of working hard at it and killing yourself, you could just go on holiday for 2 or 3 years, come back, and all the problems they'll be solved by someone else, and then you can just slot back in at a much more advanced state of development of the field. So there are 2 strategies. One is to delay getting into something until the time is ripe, and Fred Sanger did this with nucleic acid sequencing. He was studying it for 15 years before the methods, and then as soon as they were available, he jumped in, and he was then in the lead.
I am going to plug Jeff Lichtman's. I know you've listened to Eric's, so Eric Betzig's Microscopists podcast, and interestingly how he actually doesn't like electron. He was quite controversial, and he said electron microscopy is not, it's all got to be like microscopy. And then actually what you've just been saying, you then got the other extreme where Jeff Lichtman is working on looking at the brain, 3D electron microscopy through that. And it's interesting how you've got all these various aspects, but you're absolutely right. We need to do it live.
Yeah, you do.
And it needs to be there. But we need these other techniques to keep informing us so we can make sense. When we get there, that will help us make sense of your 1 micron per microsecond for the full volume. So it's not work in vain.
Well, you can do magnetic resonance imaging of the whole body and the whole brain, but the resolution is about 1 millimeter. So to go from 1 millimeter to a micron, it's 10 to the 9th. It's off the horizon at the moment. So although, Eric Betzig, they're all terribly enthusiastic about what they do, they're all just scratching at the surface, really. I mean, the power of the methods you need is just so much further in the future. I think it's, but one area that's interesting, which the synchrotron people are building X-ray microscopes now. So there is 3D X-ray tomography, and there's a fantastic video now from a group in London called Peter Lee, and they go to Grenoble, and Grenoble synchrotron has just been upgraded to, and then all the synchrotrons in the world are planning upgrades. And so it's not, you can't get angstrom resolution because of the electrons being better than X-rays, but you can do, Jennifer Lippincott-Schwartz works at Genelia on this type of plastic section embedding and so on. And they can get 20, 30, 40 nanometer, or even 2, 3, 4 nanometer. They can get resolutions subcellular that you can see organelles, but only of 1 or 2 cells. But the X-ray 3D tomography, you can do the entire body. You can do, so there's if you look at Peter Lee, who's at probably University College London, they've got a lung of a normal person and a lung of a coronavirus person, and you've got a complete X-ray, and of course it's a dead person. You can't do it live; it's dead. So you could donate your brain after you die, and then that could be analyzed by this method. And they can zoom. They can't do the whole thing at 1 micron, but they can zoom in on sub areas. They do the whole thing at low resolution, and then they can do very finely.
Yep.
So, there are things coming along, but they're still nowhere near powerful enough.
But then arguably where you started and, the innovations you've come through with over the last 30, 40, 50 years is exactly what you're saying. So the next 20, 30, 40, 50 years, it could get there. Could. There's always a way. There's always a solution. It just takes time and money to get there.
It's time and money, exactly. But one of the things that I studied in 1960 was fusion research, and I decided against it because everybody said, "Oh, it'll be 30 years." Okay, so but 1965 plus 30 years, 1995. You ask, how is fusion research getting along in 1995? And they say, "Oh, it's going to be about another 30 years." So now here we are in 2021. And they're building, fusion demonstration reactor in Japan and so on. And they say, it'll be about 30 years. And what are we saying about memory and learning? We're saying 30 years. So that basically means you can't predict it. Some things go faster. So, computers and DNA sequencing went faster than we predicted.
So in 30 years' time, we have to come back to this podcast. People have to hear you quoting that 30-year mark and go, how wise even then into that point. Richard, we are up. We ought to stop at this point because podcast. Thank you so much. I've got to say, it's been enlightening. I didn't realize you were such a wheeler dealer, which has just been amazing throughout all your career and outside. And if you are getting rid of anything, I always have a £5 note on the side.
Right.
Back to that. Richard, thank you very much for your time.
It's all right. Great fun to chat. Yeah.
So thank you everyone for listening. And if you'd like to hear more, do go to the Microscopists podcast or the Spotify, YouTube, iTunes, anything else. You can hear Eric, you can hear Jennifer. But this has just been so brilliant today. So thank you very much for listening.
Good to talk, Peter.
Thank you for listening to the Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.